Assalamu alaikum grade 12 students. Today we are going to continue chapter 2 by explaining document 2 transmission of allelic genes. Back in document 1 you have learned about all crosses described by Mendel as he worked on pea plants to study the transmission of some genetic traits from one generation of peas to another. Mendel's observations and conclusions contributed later to the understanding of the behavior of chromosomes during meiosis, by which alleles are distributed into gametes. Pea plants have many characters, but Mendel studied only one pea character at a time. Each character is coded by one gene, like the gene responsible for seed color, seed shape, or pod shape and flower position. So all crosses performed by Mendel actually studied the transmission of only one gene. Such crosses are called monohybrid crosses or monohybrids. So in this document you will learn about the segregation of chromosomes during meiosis. You will also practice factorial analysis in a monohybrid cross, and finally you will get to learn about lethal alleles. Let's start by the Mendel's first law, law of segregation. Alleles segregate randomly during meiosis. The possibility that a gamete gets a particular allele is equal to other possibilities. For example, for a hybrid gray mouse having two different alleles, gray and white, each chromosome migrates to a different cell after the separation of the homologous chromosomes during the first meiotic division, with a probability of 50% for the cell to get the gray allele and similarly for the white allele. Then, after the separation of the sister chromatids during the second meiotic division, alleles also segregate randomly, so that four gametes are obtained with 25% each, two gametes having gray allele and two gametes having white alleles. This chromosomal analysis during gamete formation is represented in document B of your books. In a chromosomal analysis, you represent alleles with letters on the chromosome, and then show how those chromosomes behave during meiosis. But sometimes you are asked to make a factorial analysis for a certain cross, like the factorial analysis of the monohybrid cross performed in document C of your books between a pure gray mouse and another pure white mouse. In the factorial analysis, you are supposed to start with the phenotype of the parents, then write down the genotype of those parents, then show how the alleles are segregated into the gametes. In this case, one type of gamete is produced by each of the pure parents. Then you are supposed to show how the union of the produced gametes gives a new offspring whose genotype and phenotype should be written as well. In this case, all mice obtained will have the same genotype and phenotype since it is a hybridization cross. But in other crosses in which parents are hybrid and produce two different types of gamete, like a self-cross, you will need to use the Punnett square table to show the different combinations of gametes to produce the different offsprings, just as in the rest of document C of your books. So, to make a factorial analysis of a self-cross, let's take the example of crossing between two F1 gray mice. First, write the phenotype using the symbol in brackets. Then, write the genotype of both parents. In this case, both parents are hybrids. Now, write the possible gametes produced by each parent. In this case, each parent produces two types of gametes with 1/2 each. Now use a Punnett square table to show the result of gamete combination in F2 generation. Write the female gametes in the first side of the table, then the male gamete in the other side of the Punnett square, then write the combination between both alleles to obtain the genotypes. In this case, 1/2 of mice are hybrid gray and 1/4 of mice are pure gray, while 1/4 are pure white mice. This is the genotypic results of F2. Phenotypically, there are 3/4 of mice gray and 1/4 are white. Those phenotypic proportions of descendants, 3/4 for the dominant phenotype and 1/4 for the recessive phenotype, characterize the self-cross in case of dominance, but these proportions are not always the same for all self-crosses. For example, if you are asked to make the necessary factorial analysis to find the phenotypic and the genotypic proportions of the flowers obtained when crossing two pink flowers of the F1 generation together, like the example of document E, page 45, what will be the obtained results? In factorial analysis, you are supposed to start with the phenotype of the F1 flowers. They are pink. You can write the word pink or the symbols inside brackets. Remember that both alleles are capitalized since it is the case of incomplete dominance, and since pink is an intermediate phenotype, then the pink flower possesses both alleles red and white. Then write the genotype. After you write the gametes, use the Punnett square table to find the results of the F2 generation. Unlike the case of dominance, you will notice that every genotype refers to a single phenotype because both alleles are incompletely expressed in a heterozygous. So, 3 phenotypes are obtained: white, red, and pink, with same proportions as the genotypic results, where there are 1/2 pink heterozygous genotype, 1/4 for the white homozygous genotype, and 1/4 for the red homozygous genotype. So, as you can see, the phenotypic proportions of descendants can be used to identify the relation between alleles. In case of dominance, there are 1/4 for the recessive phenotype and 3/4 for the dominant phenotype, while in the case of codominance or in incomplete dominance, there are 1/4 for the first parental phenotype and 1/4 for the second parental phenotype, while 1/2 for the new intermediate phenotype. Let's practice this together by studying the transmission of the coat color in the black and yellow mice, and find out what other information can we get from studying phenotypic proportions of descendants. A cross between two black mice gives only black mice. No matter how many times such a cross is repeated among black mice, the same results are obtained. What conclusion can you make from this result? This cross shows that these black mice belong to a pure race and their genotype has 2 black alleles. Now, in the second cross, 2 yellow mice are crossed. This cross gives both black and yellow mice. What conclusion can you make about the relation between both alleles, black and yellow in this case? Consider the black mouse obtained in descendants of this cross. This black mouse must have inherited at least one black allele from one parent, but this black allele is not expressed in any of the parents, which are both yellow. Then yellow allele masks the black allele. Yellow is dominant, while black is recessive. Now, what does the same results tell about the genotype of each parent? Once again, consider the black mice in descendants. Since black is just proved to be recessive, and a recessive allele is only expressed in a homozygous, then this black mouse has the genotype BB. Each of the black alleles in the genotype of this mouse is inherited from one parent, so both parents must have at least one allele B in their genotype. But both parents are yellow and must as well have at least one allele Y. Then both parents are hybrid with genotype YB. What are the expected phenotypic proportion of the descendants in this cross? You must be saying that since both parents are just proved to be hybrids, then this cross is a self-cross, and the expected results of such a cross in the case of dominance is 3/4 of the mice should have the dominant yellow phenotype, and 1/4 of the mice should have the recessive black phenotype. Good thinking. But this is not the real result of this particular cross. Referring to document F of your books, in this document you can see the proportions obtained from the second cross. Actually, this cross gives 2/3 yellow mice and 1/3 black mice. What do these proportions indicate, and why are they different than the expected ones? You expect these results 3/4 for the yellow mice and 1/4 for the black mice, but you got these results 1/3 for black mice and 2/3 for the yellow mice. If you use the Punnett square table, you will directly notice that the homozygous YY is the missing proportion, since this will give 2/3 yellow to 1/3 black mice. But why are homozygous yellow missing? For coat-colored gene in mice, yellow allele is a lethal allele. A lethal allele is an allele that leads to the death of its homozygous carrier. So, whenever you find missing proportions that you are familiar with, think of lethal alleles. The results obtained in the third cross confirm our conclusions about the yellow allele. Since the cross of the yellow mouse with a black one always gives 1/2 yellow and 1/2 black, it indicates that the yellow mouse always gives 2 gametes, one with yellow allele and the other is with allele black. Thus, yellow mice are always heterozygous, since homozygous ones die because of the lethal allele. So, I guess it is really important that you memorize all expected proportions of crosses so that you can detect any changes to these proportions, and here is a table that sums up those proportions for you.